The new chain of DNA grows by extending the 3' end of the primer. So as you have seen, in a primer template junction, one primer is present and the primer has one end which is 3' end. So the synthesis of new strand that will be continued on this side of the primer, that is on the 3' end of the primer. The phosphodiester bond which is formed between the existing nucleotide and the new coming nucleoside triphosphate. So this bond is formed in an SN2 reaction. So in SN2 reaction, the hydroxyl group of the 3' end of the primer attacks the alpha phosphoryl group of the incoming nucleoside triphosphate. So when a new DNTP or any deoxynucleoside triphosphate comes, when it comes to the site of primer template junction, the 3' end hydroxyl that attacks the alpha phosphoryl group of the incoming nucleotide triphosphate. So the after this attack, one group is leaving the reaction, and the leaving group of the reaction is pyrophosphate. So pyrophosphate, what is pyrophosphate? Pyrophosphate arises from the release of beta and gamma phosphates of the nucleoside triphosphate. So as you seen the OH of 3', it attacked the alpha phosphoryl bond. In the result of this attack, that bond was broken, and the two phosphate groups, that is the beta and gamma phosphate groups, they are released as a result of this reaction, and these two phosphates are called pyrophosphate. So in this figure you can see this is a junction, that is template and primer junction, and here you can see this is hydroxyl group, which is 3' hydroxyl group of the primer. This is a new nucleotide triphosphate which is coming to join to the nucleotide on this template. So this as you can see here, if adenine is present, so this nucleoside triphosphate containing thiamine, it comes here to be added to this growing chain. This hydroxyl attacks on this phosphate, and after attack you can see these pyrophosphate, this is released, and a new nucleoside triphosphate or nucleotide, here you can say now it is nucleotide, this is added to the primer side, and again in the next step another nucleoside triphosphate will come, and again this hydroxyl will attack on the alpha phosphate, and again the same reaction will be repeated. So in this way the primer chain grows and new DNA is synthesized. So the template strand directs the which of the 4 nucleoside triphosphates is to be added. So this template decides, and not the primer decides, which type of nucleotide will be added. So the incoming nucleoside triphosphate base pairs with the template strand, because this is the template nucleotide which will allow the incoming nucleotide to base pair with that. So what is the driving force for the addition of nucleotide to the polynucleotide chain? Because new nucleotides are continuously added, so there must be some driving force or driving energy required for this addition and for this synthesis of new molecule. So the free energy for this reaction is provided by the rapid hydrolysis of pyrophosphate, as you have seen, one byproduct pyrophosphate is released when a new nucleotide is added. So this pyrophosphate, which consists of two phosphate groups, it hydrolyze, and after it hydrolysis, two phosphate groups are produced, and the enzyme which bring about the hydrolysis of pyrophosphate is called pyrophosphatase. As you know, the bond between the two phosphate groups is high energy bond. So when this bond is hydrolyzed, a huge amount of energy is released, and this energy is used. It is the driving force for the addition of incoming nucleotides to the existing chain. The net result of nucleotide addition and pyrophosphate hydrolysis is the simultaneous breaking of two high energy phosphate bonds. These two high energy phosphate bonds are present. One is present between the alpha phosphate group and beta phosphate group. This bond is broken first, and the second bond, which is present between the beta phosphate group and gamma phosphate group, which is actually present in a pyrophosphate. This is again hydrolyzed. So the hydrolysis of these two high energy bonds, they release much energy which drives this synthesis. So as you can see, this is a primer template junction, and when a nucleotide triphosphate comes here, it is added to the 3' side of the primer, and two phosphates, which is a pyrophosphate. This whole molecule is pyrophosphate. This pyrophosphate is released, and in the presence of an enzyme pyrophosphatase, this bond, which is a high energy bond, this bond is broken, and two phosphate groups are released. So you can see in this diagram that whenever a new nucleotide is added, two phosphates are also released. So this is the net reaction at the 3' side of the primer template junction. Therefore, DNA synthesis is a coupled process. This synthesis not only consists of a single reaction, that is a nucleotide has come and it has joined with the template nucleotide. Rather, another reaction, which is the hydrolysis of pyrophosphate. So these two reactions go on side by side. So this is a coupled process. This reaction, another significance of this reaction is that this reaction is highly favorable with high value of equilibrium constant, which means that it is an irreversible reaction. As you have seen, the events which have taken place for the addition of a nucleotide to the primer and template junction, these events cannot be easily reversed, because it has high value of equilibrium constant. So this reaction is irreversible.